My name is Ronnie Green, and I have the pleasure of serving as the University of Nebraska-Lincoln's Vice Chancellor for the Institute of Ag and Natural Resources. Welcome this afternoon to our second Heuermann Lecture series event. We started this event last month in October and kicked it off with Professor Swaminathan from India, the first winner of the World Food Prize, as our guest. And this afternoon, we're very pleased to bring the second Heuermann Lecture to you with Dr. Stephen Baenziger. Our intent in this new lecture series is to bring to our campus and to our community at UNL and for our greater stakeholders in the area and the state discussions centered on the issues around food security, around natural resources security, around energy security, food, fuel, and water, so to speak, if you wish to think of it that way, and the sustainable rural landscapes that are going to be required to meet those challenges for the world in the future. We have been able to start this effort this year on a monthly basis, every month during the academic year and into the future, from the generosity of a gift made to the university by two long-term supporters of the University of Nebraska, Keith and Norma Heuermann. The Heuermanns are leaders in Nebraska agriculture, have been throughout their life. They're from Phillips, Nebraska, about an hour west of us here in Lincoln, and we're very, very pleased that Keith and Norma are here with us again today. If you would join me in welcoming them and thanking them for their generosity. Now, there's always a little danger in introducing a few dignitaries that are with us, but I will point out a few that I saw come in. We're very pleased that President Milliken, the President of the University of Nebraska, is here with us this afternoon. Regent Jim McClurg from the Board of Regents is also here with us. Thanks, Jim, for being here. Lieutenant Governor Rick Sheehy for the state of Nebraska is here on the front row with us. Thank you, Lieutenant Governor, for being with us. And our Director of Agriculture for the state of Nebraska, Greg Ivaugh, is also here on the front row. So thank you for being with us this afternoon. We also have a number of special guests with us, and I'm just going to ask them to stand and be recognized as a group, if you will, who are with us because tonight our guest speaker will be bestowed the Nebraska Wheat Growers Presidential Chair by the University of Nebraska and the University of Nebraska Foundation. And as part of that is able to happen because of a new licensing agreement that we have with Bayer CropScience through NUtech Ventures and with the university and our partnership long time with the Nebraska wheat industry. So we have a number of representatives here from the leadership at Bayer CropScience. I'd ask you to stand if you would. Join me in welcoming them. Thank you for your partnership. And we also have with us several members of the Board of Directors of the Nebraska Wheat Growers Association. So would that group please stand and be recognized where you are? They're still in their meeting. Okay. They're going to be here, Steve, to hear you. The lecture this afternoon is being streamed, web streamed, and will be archived on the web at heuermannlectures.unl.edu. So we're joined with folks from around the state and the region in the audience with us. Our lecturer this afternoon is no stranger to many of us here on the University of Nebraska-Lincoln campus. As I mentioned, Dr. Stephen Baenziger will be the holder of the first Nebraska Wheat Growers Presidential Chair at the University of Nebraska. He is a small grains breeder, specializing in work in wheat, barley, and triticale in our Department of Agronomy and Horticulture, where he has been a faculty member since 1986. Steve is a native of Toronto, Canada, which he refers to often in his background. He holds a BA degree in biochemical sciences from Harvard University, where he graduated magna cum laude, and a master's and PhD degree in plant breeding and genetics from Purdue University. He has worked since his joining the faculty those number of years ago, especially in the wheat breeding and improvement area to improve that crop as well as the other small grains in order to feed the world. As a measure of the reach of his work, in the last 5 years, he and his team have released 6 new wheat cultivars, and the reach of that extends to over 65% of Nebraska's wheat acres currently in production. He's one of only 2 Americans that serve on the Board of Trustees of the prestigious International Rice Research Institute known as IRRI, and he has, in his own words, described that appointment as one of those 2 Americans to be a dream come true for him. In March of 2011, Steve was awarded the Outstanding Research and Creativity Award, the ORCA Award, by the University of Nebraska, and it is, as you know, for those of you that are university folks in the audience, the university's top award for research creativity. Other recognitions that he has received include honoree in the Nebraska Hall of Ag Achievement, the Nebraska Agribusiness Club's Public Service Award that he received last year, the CropScience Scientific Society of America Research Award, the American Society of Agronomy's Agronomic Achievement Award, amongst many others. One of the things that I expect that you will hear from Steve this afternoon, and one of the reasons that we thought he was such a fitting one of our kickoff lecturers for the Heuermann series, is that whenever he is recognized, he always makes a strong point that he is receiving that recognition on the behalf of others. The people that he works with in his teams, the people that do the work on a day-to-day basis, that make that reach of his work so strong. He's an amazingly generous person and servant. Steve will tell you that if you're going to work on something you believe in, and feeding people is what he says he believes in, that has always motivated him. So I look forward, Steve, and welcome to the podium to deliver the Heuermann Lectures on Setting the Stage, Why Agriculture. Please join me in welcoming Dr. Stephen Baenziger.
It's a great pleasure to be here, and I want to thank the organizers for first of all inviting me. I want to thank Keith and Norma Heuermann for helping this lecture series get started. And as we said, let's see if I can do this. The title of my talk will be Setting the Stage, Why Agriculture. And what I wanted to do in this talk is to sort of put it in a Nebraska perspective. We're going to have many global speakers here, and I think it's important to recognize the important role that Nebraska fits within that system. And just to make sure everybody's in the right room, I always like this slide. No man qualifies as a statesman who is entirely ignorant of the problems of wheat. Socrates. Okay? So we're all in the right room, we're in the right mindset now? Okay? Just making sure. That was said about 2,500 years ago, and it talks about the importance of agriculture. And I think one of the themes that will come out today is that agriculture is as important today as it probably ever has been in the history of humankind. And the topics we're going to talk a little bit about is who we are, and this gets, I think Ronnie may have looked at my slides almost, in that we're going to talk about who the team is, what we do, the challenge. And I am so pleased that there's a seminar series of this nature on agriculture because of what agriculture means, and it could not have happened at a better time as we look forward. We're going to talk about what plant breeding is as an integrative science and how it's evolving. And I'll try to make it so we can all understand it. Normally, if I can understand it, everybody in the audience can too. This is the team, and it's large. We have a number of people that work in genetics. We have some tremendous people working in plant pathology, variety testing, plant transformation, entomology, cropping systems. If you work in wheat, you have to understand end-use quality, so we have a tremendous group working in end-use quality. We do a lot of work with biometry and statistics, and I'll try to explain how this team all works together as we go through this. We also have modeling And then, as I mentioned, I work on triticale. I work on barley, mainly with my graduate students. And my technical support I always like to acknowledge because they keep me honest, and you know, frankly, there's not a variety that we release that isn't touched by the hand of my technicians or technologists, a graduate student, or an undergraduate. And so we owe them a huge debt of thanks. Our funding sources are mainly, of course, the university, the state of Nebraska. I want to specifically acknowledge the Nebraska Wheat Board. They're sort of the one that you know, the one you brung to the dance and you go home with at the end of the day, okay? They funded my program for every year that I've been here. And we have now, of course, R&D fees. We have endowments for which we wish to thank Bayer, and a number of other ones. Now, literally, I have drunk from wells I did not dig. I've been warmed by fires that I did not build. What I want you to understand is that agriculture is too important to be looked at as any one person. And as Ronnie actually said, I'm representing the team when I'm giving this talk. And I also want you to understand that agriculture is team research where failure cannot be an option unless you're willing to accept starvation. And most of us, I hope, are unwilling to do that. Now, let's talk about the impact. I've shown you how large the team is. In 2010, the state of Nebraska produced just under 4 billion pounds of wheat. That crop was worth a little over $500 million. Now, as a geneticist, I can't take credit for that, okay? Because that's due to the management system as well as the genetics involved. But the genetic improvement since 1966 is at least 20%. That's actually probably a low estimate, which would mean of that $500 million, $107 million at the farm gate was added by research that's been done by plant geneticists. Now, I can't take full credit for that because my varieties aren't grown exclusively in the state, but 66% of the state is in the varieties that we've released. So if you take that 107 million, roughly $71 million of increased revenue has been added to our farmers when they sell their crop due to what the team does. We think that the team cost the university and the federal government, because we have tremendous collaboration with the USDA ARS, somewhere between $3 and $4 million. And so we think a return of roughly 20 to 1 is probably something you would accept on any of your bank accounts. I mean, you go to your banker and say, I'd like 20 to 1 at the end of a year, don't you? Okay. Now, I like our farmers being prosperous. And by the way, that doesn't include that our varieties don't know state lines. They're widely grown in South Dakota. They're widely grown in Colorado, Kansas, Wyoming, and a little bit in North Dakota and a few other places. But as much as I like our farmers being prosperous, paying taxes because they support the university through their taxes, and we're, as a university faculty member, immensely proud of that aspect, that's not why I got into agriculture. Why I got into agriculture is this middle column. That same wheat crop, if you looked at the annual consumption of wheat per person, would feed a little over 20 million people. That's roughly 10 times the state's population. If you look at how much of that's due to genetics, that's roughly 4 million people. And how much is due to the program at the University of Nebraska, it's 2.7 million people. And if you wonder why I get up every morning and come to work and why I sleep well at night, it's because I'm doing what I love. 2.7 million people are getting fed because the University of Nebraska and the USDA are allowing us to do what we like. That's what motivates us. Now, I want to bring with this, because a little bit I want to give you a history, because I think we're about on the verge where we're going to repeat history. When I was 18, that was 1968, so you can figure out the math, I'm 60 years old. There was a book that came out by Paul Ehrlich, and it was called The Population Bomb, member of the National Academy of Sciences. And what he said was, the battle to feed all humanity is over. In the 1970s, the world will undergo famines. Hundreds of millions of people are going to starve to death in spite of any crash programs embarked upon now. 18 years old, amidst the Vietnam War, amidst the civil rights movement, and you read that we're going to watch hundreds of millions of people starve, despite anything you can do. That led to a lifeboat response, which was, with starvation looming and there's nothing you can do about it, the proper thing to do we decide who's in the lifeboat and who gets to drown, okay? Who would be in and who would be out. And so, frankly, at the time, whole countries were expected to have massive, unavoidable, and because it was unavoidable, totally acceptable human losses on a scale that we hadn't seen since the plagues, okay? That's what people thought about agriculture in 1968 and its capabilities to feed people. And it's not by chance, and I really appreciate being the second speaker following Dr. Swaminathan, who is the Green Revolution father in India, where many of these losses were expected. Now, there's a contrary view, and I always like this one, and I'm going to give you 2 slides. With monotonous regularity, apparently competent men have laid down the law about what is technically possible or impossible, and have been proved utterly wrong, sometimes while the ink was scarcely dry from their pens. On careful analysis, it appears that these debacles fall into 2 classes, which I will call failure of nerve and failures of imagination. The contrary view is that of Gandhi. You can be the change you want to see in the world, that you don't have to accept your future. And that of Albert Einstein, who said the highest intelligence of mankind is not reason, but vision. And so while Paul Ehrlich was writing his book, winning prizes, selling, it was a bestseller, there were 2 people working quietly in the dusty corners of what agriculture and development world is like. One was Norman Borlaug, one was Henry Beecher. We know him as Hank. He's an alumnus of this university. Norm developed the Green Revolution wheats, and Hank was the developer of the Green Revolution rices, 2 of the 3 major staple crops that feed the world, the 2 crops that feed the poorest of the poor by far. And what they did is they created a set of varieties that when properly managed, and again, you can never separate the genetics from the management. They needed water and they needed fertilizer, but they could take water and fertilizer so they increased yield. And they changed agriculture for the next 40 years. Now, Norm won the Nobel Prize in 1972. And what he said at that time was, don't think of this as the solution. Don't think of this as the answer. Because all we've done is we've bought time. And for the next 25 to 30 years, we'll be okay. And then we'll be back to where we were. And I think he was off by about 10 years, but we're back to where we were. And so the question is, are we back to 1970, and do we need a second green revolution? That was the topic of your first speaker, and it's one that I totally agree with. Now, the challenges of agriculture. The first one is population. We've all heard about the 9 billion people supposed to be here in 2043. Reached 7 billion, they believe, last week. 10 billion, which they didn't think we were going to actually reach. They've now upped the total population demographics in 2083. Population is one driver. The other driver, which is interesting, is increased wealth. Interestingly, the 9 billion people we expect to have in 2043 won't eat like 9 billion people today because they're hopefully wealthier, which means that they're going to eat like 12 billion people today. So that puts us in a bit of a box because your increases come not only from population, but they also come from wealth, and no one is in favor of poverty. And just to put it in perspective, there's some things you can see here. For each 1% increase in real income, the demand for wheat goes up by 0.5%. The utilization of wheat is 2/3 due to population, 1/3 due to income growth. And we haven't even talked about the animal industry, which people want more protein. They want better quality proteins. And so that's going to do quite a bit. Nor have we talked about the bio-based economies, renewable resources, and nor have we even mentioned climate change. Those are the challenges. Now, on top of that, one of the things that's interesting to me is that this quote, and it was right after Norman Borlaug won his Nobel Prize, that few scientists think of agriculture as the chief or model science. Many indeed do not consider it a science at all. Yet it was the first science that makes all human life possible, and it may be well that before the century is over, the success or failure of science as a whole will be judged by the success or failure of agriculture. And I think if you look at, that was written in the 20th century, before the Green Revolution fully had its impact. I think that's probably truer in the 21st century. I think you're going to see the very best minds in the future will be coming to agriculture because we are the ones that are going to need the greatest impact and to have that greatest ability to change human life. Now, putting this into a perspective, there's an equation that every plant breeder uses. And what it is the phenotype, P, which is what you see. That's the plant physically. It's what you harvest for yield. It's how tall the plant is if you're cutting it for biomass, these types of things. The phenotype is due to the genotype. Now, the genotype is what breeders work with. We work with genetics. The environment. Now, the environment needs to be broken into two parts. One is the weather, that's uncontrollable. And the other part is management, which includes the tillage, the use of pesticides, the use of irrigation, the use of fertilizer, and the prior cropping history. Those are determined by the producer. Those are controllable. And then you have the genotype by environmental interaction, which is how do varieties respond to water, how do they respond to fertilizer, things like that. Now, I bring this up to make 2 points. The first one is that if anyone doubts that plant breeding or agriculture is in fact an environmental science, they're sorely mistaken. Everything we do is in an environmental context, first point. The second point, as we go forward, where are the gains going to be made? And this is where I think we have to be very careful. If you think about the 20th century, what changed agriculture? Synthetic fertilizers, cheap nitrogen, the discovery of herbicides where you could control weeds, the discovery of pesticides such as insecticides where you can control insects, the discovery of fungicides where you can control disease, the tremendous irrigation systems and the efficient use of water, and of course all of our equipment that allows us to do the tillage. But if your fields are insect-free, disease-free, weed-free, you have proper nitrogen and proper irrigation, and you go to that equation, what's left or what will become more important in the future, it's the one thing we can still change, which is genetics. You know, you're not going to get any more efficient. I mean, you can get more efficient and more productive by better chemistry, but once you've got disease-free, it's a matter of productivity, but you're never going to get more disease-free than disease-free. You're never going to get more weed-free than weed-free. So what it says is that genetics is going to have to become more important as we go forward. And if you look at the balance of that phenotype between genetics and the environment, the genetic scale will become increasingly important. Now, what does plant breeding do? There are 3 phases. The first phase is to introduce variation. The second phase is to allow that variation to separate out and then select the useful variants, because you'll have a lot of trashy variants that are no value. And then finally, you're going to have the evaluation and release. It takes 1 to 2 years to introduce the variation, year 3 to year 6 to do the segregation and selection, 7 to year 12 to do the evaluation and release. For winter wheat, and in summer annuals you can do it more quickly, but in winter wheat it's 12 years from start to finish. And I want to mention the very importance of continuity. If you change breeders, I mean, I've been here 25 years and I've turned my germplasm over twice. Okay? Hope to be here another 8 years, so maybe I'll get 3 times. Okay? But that's what you have to work with. Now, normally we introduce variation. We do it through pollination. This is emasculating it. These are the female parts of a plant, look like moth antenna. That's what we pollinate. And the genetic resources are anything you can cross. It becomes basically what a species is. That's the definition, what can cross. Now, in the allied sciences and what's coming on, we're looking at things like transgenes. You'll never hear me use the term genetically modified organism, and they're well, except when I have to because other people understand it. I genetically modify every organism I breed. I just do it on 30,000 genes at a time, okay? So to me, when I look at transgenes or genetically engineered crops or what other people call genetically modified, it's transgenes or traits. We also use wide hybrids and we use mutations. Now, let's take a look at how, and by the way, traits or transgenes, there's 2 great frontiers left in improving wheat. One would be transgenics, and there is no commercial transgenic wheat available today. We hope that'll change. And that's because of consumer acceptance. And the last one will be hybrids, and I'll talk about that at the end. The first frontier, transgenic crops. There's a really nasty pest of corn. It's the corn rootworm. That happens to be the larval stage, and I hope for the entomologists in the audience I'm doing this rightly. That's this guy, feeds on the roots and hurts and stunts the plant. The adult stage is in fact a beetle. Now normal corn, or the ancient corn, I should say put this way, the corn rootworm can be controlled by a nematode that feeds on the corn rootworm. And corn has the ability to release a biochemical signal that says to the nematode, come near me because you might find food. But the problem is that modern corn, by accident, has bred out that signal. So no longer does the nematode know that it should be looking for corn or finding where the corn is so that it can eat on what eats the corn. That means there's two ways you can do this. You can go back to the ancient corns and cross it in. That's time consuming, okay? Or you can transform corn with a gene that emits that signal. And interestingly enough, they went to oregano. I mean, you got to love these guys, right? They went to oregano and they said, oh, let's find a gene in oregano that actually makes this signal. They put that into corn, and then they monitored how many of the larvae grew to be beetles. So that would be how many were successfully fed on the roots and grew. And they reduced it 60% by just putting that signal back into corn. So you think about it. They take a gene from oregano. Now, most of us aren't fearful of eating oregano, right? Okay, so you could do that. It's probably, if it were, it's probably not the allergen anyway, if you were allergic to it. It's not the pesticide itself. It, in fact, calls in a biological control agent, which then feeds on the corn rootworm, kills the corn rootworm, the corn's happy, the nematode's happy, and there's no pesticides in the environment. Not a bad system. A lot of this technology gets criticized because it's believed that it's only for the developing nations. This slide shows you the adoption of transgenic plants. This shows you the adoption in the developed world. This shows you the adoption in the developing world. They're virtually the same, and it wouldn't surprise me if in the future the developing world grows more transgenic crops than does the developed world. And what's particularly important, and I like to point this out for my friends from Europe, this is the map of where all the crops are grown. By far, the United States is the major user. We have roughly half the world's transgenic crops. But there's a little country in Africa called Burkina Faso. Okay? Most of you probably don't know much about Burkina Faso. Frankly, neither do I. But in all the countries that grow transgenic crops, Burkina Faso is the number 15th largest producer of transgenic crops, and it has more transgenic crops than any country in Europe. So if you think about technology which can be used in developing countries as well as in developed countries, that's not a bad thing to remember. Now, I don't know how the Europeans expect to compete with Burkina Faso and the agricultural giant that country's going to be, but I would be wondering. Now, we also get variation through mutations, as I said. I want to point this one out for a reason. This is a chromosome arm of rye that is transferred to wheat. It gives us drought tolerance, heat tolerance, an insect tolerance, and a disease tolerance. The other reason I pointed out is that amount of DNA is larger than the Arabidopsis genome. So what we're saying is I can transfer, without any regulation, more DNA than the smallest flowering plant's genome, and nobody cares. But my gosh, if I take a sliver of DNA that would be the equivalent of a transgene, you got to regulate that. Okay? Now, I'm not saying it shouldn't be regulated, but it is interesting on perspective. Wheat is normally sensitive to herbicides. There is mutations which allow it to be resistant. This is the normal wheat. This is the mutated wheat that has the herbicide resistance. Extremely effective and very popular. So not everything in breeding has to rely on traits. There's lots of ways of introducing variation beyond that. Do want to talk a little bit about molecular markers. All I want to say is, if you look at this, you can see some bands are down, some bands are up. These happen to be simple sequence repeats. And all they're doing is they're giving us a street address in the genome. And I'll explain that in a minute. We did this because the first study is to understand the variation, that first phase. What we knew is that the wheats of the Great Plains were introduced from Turkey. Turkey Red was the founder wheat that changed wheat agriculture, okay? And in its day, it was the most widely grown wheat. It was grown basically from the Canadian border to Texas. Came from Turkey. It had the heat stress, the drought tolerance, and this, what we would call a steppe wheat from the Russian steppes that could do it. It also was a bread wheat. Before that, we grew in Nebraska spring wheats and in the southern Great Plains soft wheats. So the idea is Turkey wheat came to the U.S., became wildly popular. It was brought, by the way, by Mennonites. And this happens to be a statue to the Mennonites bringing it in. And some of the members in our audience are actually in this picture. I don't know if Casey's smiling or not, but she's there. And this is in Newton, Kansas, where they actually brought it in. Now, the idea then became, if wheat for the Great Plains originated in Turkey, and we brought that wheat over in 1874, and we've had a breeding program in Nebraska since probably the 1900s, and we've gone our way and Turkey's gone its way, because they've had a breeding program ever since 1924, effectively, maybe there's still genes that we could find that belong together. So could we go back to our homeland, basically Turkey, to find new genes to bring them back in? We used molecular markers to do that. Most of the Turkish wheats, which are in black, clustered by themselves. Most of the Great Plains wheats clustered by themselves. But Turkey brought it in 1874. Cheyenne, the first wheat that was released by the University of Nebraska of any significance, very similarly related to it. Siouxland released in 1984, a very major wheat, still highly related to that. And we see in this cluster a number of Turkish wheats that are related to our Nebraska wheats. We see down here a few Turkish wheats related to our Nebraska wheats. And so now we know how to go back systematically and sample germplasm to create parents and to create new lines. We're going to go back to our homeland to get those kinds of materials. Now, after we've introduced variation, now we have to do segregation and selection. There are 4 traits that every variety in Nebraska must have. It has to survive the winter. It's totally acceptable if it dies in my breeding nurseries. There's not a grower I know that wants to see a dead field in his farm. Okay? So we've got to get rid of those. This is stem rust. It's also known as black rust. For those who are familiar with the biblical plagues, the 7 plagues of Egypt, this is the rust that they refer to. Devastating disease. Some of you may know of UG99. It's the same disease. Has to have agronomics. It has to yield. It has to stand. It has to be harvestable. Those types of things shouldn't shatter out. And when you work with wheat, it better make a loaf of bread or make a noodle. Those are our 4 traits. In order to identify those traits, we have to test clear across the state. And what I really like about Nebraska is this is basically an evapotranspiration map. You can see this is where most of the water is. This is where it's much drier. In the east, you're predominantly corn and soybeans with a little wheat thrown in. As you get into the drier section, sorghum replaces the corn. As you get into the much drier section, wheat becomes the dominant crop. Where you can't grow wheat, you grow grass. Importantly, from Omaha to Scotts Bluff, there's as much climatic variation as there is from Omaha all the way to the Washington, D.C. So what it means is that we have, when it comes to that phenotype and that G by E, this is the cauldron. If you've ever looked at the weatherman, when he's not standing in front of Nebraska, which they often do, all the isotherms seem to converge on our state. And that's part of the deal. But this is where we do our selection nurseries. A lot of that's done with the traditional, plots and harvest. But we're also evolving to better assays, marker-assisted selection, things like doubled haploidy. Now, most people think everything has to be done with genomics. Doesn't. This happens to be an optical scanner. Anything you can use light to separate seed on, this will do. Now, when we say light, it doesn't have to be visible light. It can be NIR, it can be UV. We use this to separate red kernels from white kernels. You can use it to separate diseased kernels from resistant kernels, or plants that are grown on resistant plants. You can use it, perhaps, if we're really careful, how to separate on protein levels, which are very important for human nutrition as well as for grain grading. Non-destructive, and you can do literally thousands of kernels in a very short period of time. We have this is a prototype, we have a standard one actually made in Lincoln, Nebraska, and it literally does hundreds and, well, thousands of kernels at a time non-destructively. Again, we do a lot of work with molecular markers. We can determine which chromosomes have recombinants, various forms. The important part is that these become like street assays. So A means it came from one parent, B means it came from another. If you discover every line that has an A here has a trait, then you know exactly where it is in the genome. So we found things like this. This is a very interesting part on a chromosome in wheat, chromosome 3. This happens to be a score for a trait that affects yield. At this location, and again we have markers along this chromosome, basically street addresses on a street. This allows us to find traits that are 15% higher in yield than if the trait weren't there. What's interesting, that's the trait for the allele from Wichita being put into Cheyenne. If you then take the Cheyenne allele and put it in Wichita, it reduces yield by 15%. So you have the mirror image, and you're looking at 15% yield, which is huge. You know, a new variety is often 3, 4, 5% better in yield, never 15%. What's even more interesting, and I want to explain this curve, this is an environmental index, which says we're in low-yielding environments here, high-yielding environments here. This happens to be the allele, the favorable allele, and this happens to be the Cheyenne allele, which is less favorable. What you see is in no environment does the Cheyenne allele ever hurt you. Or the Wichita allele. The Wichita allele, the purple, is always higher than the Cheyenne allele everywhere. It means that you can put this allele in and it never hurts you. It's beneficial everywhere. Now, that's not always the case. This happens to be for a gene for height, an allele for height. In this case, the Wichita allele, when you're in short environments, when you want to grow tall plants, say for biomass, you want it. But as you get into better environments where the plants are taller, then the Cheyenne allele becomes better. It's a crossover is what they call it. The problem with that is, how do you predict at planting which one you're going to need? Can any of us say if we're going to have a dry year or a wet year? And so how do I make a recommendation to a grower as to, well, grow this one if it's going to be a dry year, thanks. Grow this one if it's going to be a wet year. You can't do it. So the concept of having a gene which is uniformly better, never hurts you, is very, very important. Where is it optimally best? In the high rainfall areas. This is the yield gene. That means it's going to be best in these areas. Now, one of the things we were interested in was if you could find a trait that would increase yield by 15%, you'd like to kind of believe as a breeder you were able to use it, right? You don't want to be thinking that as you're making all these combinations that something that's that big we just couldn't find. Our selection protocols didn't work. So what we did is we took a series of lines that were historic to modern to see which allele did they have. Now what we found was the Cheyenne allele was still present. We were puzzled by that for a little bit. The Wichita allele was also widely used. And of course when you make crosses you get new alleles. But when we started to look at the lines that clustered that had the Cheyenne allele, what we found, was there were varieties, with the exception of one, that were adapted to this area. Now, if you remember, that's the lower yielding area, and that's where the allele had virtually no differences, okay? That would be this area. Yeah, it was better, but not very much better. So our selection technique wouldn't find it. And where we found the lines that did have the Wichita allele, they were almost exclusively used in either the high rainfall, eastern Nebraska, or the very best wheats in western Nebraska for irrigation. And so even though the Cheyenne allele is still present, it's present only in those varieties where it had little effect, that are grown in areas where the allele had little effect. Now, marker-assisted selection, I want to give you an example. And I hope you can see this. Where this orange line is where the flood line was. I don't know if you can look at the trees, but they have these clean stems, clean parts here. And then down here, it looks like that, nothing but roots. That, in fact, is when the flood came, it took all the debris from the field, that's all basically grass, and it went right across those, the stems of the trees. And as the floods receded, they left the grass there. So this is all basically flood debris. There's a gene that they found in rice called submergence tolerance. Now, rice is grown in water. We all know that. But what you don't know is it will drown if it's completely submerged. And you only need to leave it underwater for 7 to 10 days. But with the new submergence tolerance gene, it can be underwater for 7 days, basically goes dormant, and it'll survive. So here we are in one of the poorest parts of India. That's the flood line. This is a variety of rice that had the gene. This is what it looks like. This is a field of rice that didn't have the gene, completely died. It's now planted to vegetables because they're trying to capture some kind of crop. You may ask, well, are they stupid? Why didn't they grow Swarma sub 1 on both sides of it? It was lack of seed. They're ramping it up. They just don't have enough seed. Every village is trying to get as much as they can, but it takes time. And then these are the people that we got to meet. Basically, this village is going to have rice this summer because they will be able to get a crop from that. And that was all done with marker-assisted selection. So if you wonder how important that can be and what it means in real people's lives, just take a look at this field and that field. Now, that's basically going into marker-assisted selection. It's evolving into marker-assisted breeding, and it'll eventually go into genome-wide selection. Tools that I'm not going to spend a lot of time on, but I do want to show you a little bit about how we use it. I have a very good graduate student. He looked at our intermediate populations. We put roughly 2,000 markers on them. We wanted to see what kind of diversity do we still have in our nurseries. We found we had 3 main clusters. Okay, so we're fairly diverse. That made sense. We then went in and we selected the lines, and we wanted to see how our selection worked. What we discovered is we selected nothing from the smallest cluster, which was interesting because by chance we selected enough lines that we should have gotten 1 or 2 from there. Most of them came from this cluster, most of them came from this cluster, and now we're going to go back and start learning how to maintain diversity within our populations because, what did we say? The first phase of breeding is putting variation in. We've got to make sure we keep it. We don't want to narrow it too quickly. And that leads us to one of the neat parts about being a plant breeder and being an agricultural scientist and a scientist in general is that we shall not cease from exploration, and at the end of all our exploring will be to arrive where we started and know that place for the first time. I have selected since I was 24 years old. I've done it. We've been successful. 66% of the varieties in the state are in our materials, okay? We never knew exactly how we were handling our diversity until now. We're able to do that, and so for the first time, we're beginning to really tease apart our breeding systems so that we can be much more efficient. Of course, we still run a lot of plots. We still make a lot of loaf of breads, by the way. That's a commercial control. That's one of our experimental lines. That's one that the quality lab said, please never let it grace our door again. Okay? And that happens. Okay. Now, the last part that we have to work with is cropping systems and evaluation, and does it need to be released? And this is where I really want to acknowledge the efforts of our statistics department and our crop modeling and the people we work with in crop production. You know, we have a major effort in water for food. If you wonder what a field that looks like under drought, this is it. You can see this part was droughted out. Now, that's a uniform stand of wheat, okay? And I want you to think about it. I make my career separating genetic differences. Same variety. How do I separate out this thin plot, this non-existent plot, this partially existent plot from this plot, fully there? That's all field variation. I go to my friends in statistics, and they help me out. This happens to be, we told you about winter hardiness. You see the snow bands. Wherever you see a snow band, that wheat will have less winter killing because snow works like an insulation. And once the snow melts, it'll have more moisture. So you see how nice and uniform, how it clearly follows your replicates in the field? You know, I mean, it's a totally regular and defined shape, right? Okay? How do we take that out? We take it out with statistics. Now, there are 3 joys that you'll find in plant breeding, okay? The first one is when you make the cross. That's when you take the variation, you put it together, and you think about all the cool things that can happen, okay? The second is when you go out and you see it in the field and you see a line that you know will be a variety. You'll remember the time of day, you'll remember where the sun was shining, whether it was windy, you know, where it was in the field. And the last time is when that variety is grown on millions of acres. And you remember when you held all the seed of it in the palm of your hand. And as a scientist, you have to remember, and as an agricultural scientist specifically, what you hold in the palm of your hand. Because we change people's lives. Norm Borlaug, there's 1 billion people today that are alive because of what he did. That's not a bad epitaph. Now, want to go to this next slide. By the way, if you wonder how to have profitable wheat production, okay, and that's the way it works, right? You know? I mean, that's your insurance policy, shall we say, okay? But it comes down to this. A good past is positively dangerous if it makes us content with the present and unprepared for the future. And in agriculture, the thing that worries me the most, and frankly, one thing that I personally hate is when a breeder describes this program as a numbers game, as if you can just run enough numbers and you're going to be successful. We never can be complacent. And so we always have to be prepared for what the future brings. Now, I've talked to you about traits. I want to give you the final frontier, which is hybrid wheat. Now, this is something which they tried in the 70s, didn't work particularly well. They're going to try it again, hopefully now. It's grown in India, it's grown in South Africa, it's grown in Australia. These slides, by the way, are from Mexico, which is a company founded by Mr. Bawali, a friend of the university, and a very fine gentleman, first ethical seed company in India, and also a World Food Prize laureate. The problem with this technique is that the seed costs are very high. It's difficult to make a self-pollinated crop, a cross-pollinated crop. And then we also, because wheat is a polyploid, not that you need to worry about that, but heterosis tends to be lower than in maize or rice. There is lots of efforts in how to develop hybrids. They've been somewhat successful. The chemical hybridizing agents, the CHAs, have worked. The better one appears to be cytoplasmic male sterility, mainly because it's more efficacious and easier to use. The biggest problem has been restoration. By the way, I should say, in hybrid rice, it's now grown on millions of hectares. So how did Mexico approach this problem? It said, first of all, we have to do all our testing at half the seeding rate. That'll cut the seed costs. And so that's how they're doing the tests. Hybrid wheat in Australia, or not in Australia, but in South Africa, they plant 5 pounds to the acre, which is basically 1/10 to 1/12 of what we plant. That makes the multiplier work. They do it with very precision air seeders, and it's very successful. They have their cytoplasmic male sterility. Genetically, I don't know if you can see it, but they put in a multiple ovary in the seed. Usually each flower has only 1 seed. This has 3. They're hoping that if the pollination can occur, that pollination will get 3 seeds instead of 1. That greatly reduces the seed cost also. Now, the interesting part is nobody could mill that grain. But that trait is recessive so that in the F1, you're going to grow plump, large seeds that every miller is going to like. This is how they do their experimental hybrids. Their production fields, in India, they're small farms. That's a male row. 6 feet away is another male row. In between is the female row. There's lots of pollinators to pollinate that. Clearly, we couldn't do that on any scale in America because we need much larger equipment. But that's how they're approaching it. And I think it's not by chance that this technology will take the patience that only a developing country has. And that's basically how hybrid rice was developed also. But we're hoping we can add something to this because it is one of the 2 great frontiers for wheat. Now, there's an old African proverb which says if you want to go fast, go alone. If you want to go far, go together. And I think if you look at it, we have 9 billion people. Whoops. Pardon me. Okay, now I'm really embarrassed on this one, JB and Ronnie. Let me go back. That N was supposed to be the end of billion, and I don't know how it happened when we put it on this thing. But just imagine a billion with the B from bear and the N from Nebraska. Yeah, okay? Just remember that, okay? Visualize that, you know? And what it says is that we all have to work together, okay? I guess today I'm a little bit, if we had to make a mistake, it's probably best that the N was missing. You'd cut some slack on that one? Okay. Now, with that, I'd like to say, come on home, Ann. Thank you. And I hope you've had a good time. I hope you've understand, A, the importance of agriculture, B, that it's only done as a team research. I'm honored to be here as a member of the team. I acknowledge all of my fellow people that work on that, and that I think the importance of agriculture is only going to get more important. And I really wish to thank the sponsors of this symposium, Keith and Norma Heuermann, for sponsoring this. Thank you.
Okay, I think we have mics on the floor. Jill and Mary are going to help us. If there are questions from the floor that we'll take for a few minutes from Dr. Baenziger. Are there questions? Mary is in the back with a mic and Jill is over on the side with a microphone. Questions for Steve? Right over here, Keith. You probably got a loud enough voice, Keith. You can just do it on your own, huh?
Thank you, Dr. Baenziger, for the presentation. I can understand it. I'm just a farmer out there, so I appreciate it. Question I have. I am Keith Olson for the people that don't know me. Farm out in southwest Nebraska, wheat, dryland farm, 100% no-till. We hear so much in today's society that we don't need to use transgenic crops. We can raise enough organically, naturally grown food to feed the world. You start out with the indication that we couldn't do that. Is there a way that we can somehow tell the story of what's going to take to tell to feed the world? You know, we believe that we need to feed the world, although there's people out there said we don't need to feed the world. It's a frustrating situation to be in because we believe that feeding the world is what we're here for. And yet there's people that are challenging us in that statement. Just comments.
Yeah, I think the difficulty always is, and it relates to something which is far bigger than agriculture, it relates to scientific education and the process of how you would use the technology. You know, I personally believe, and frankly, I believe the transgenic crops are going to be a huge tool, but I could believe that others would disagree. But the point is, I don't want them to disagree for non-scientific reasons. But again, if you look at the culture that we live in, we're permeated by non-scientific reasons. I mean, you can laugh at the French who don't like transgenic crops, but they have 88% of their power is nuclear power. They're contributing far less to greenhouse gases than we are. And so, we irradiate meat and we think we're all going to glow in the dark. Well, other people don't think that. I think what you need is a scientific literacy, and unfortunately science itself has become more political than what it should be. But I think food will probably trump it. The first transgenic rice is going to be golden rice. It's going to be, and I didn't mention this, but there's 2 billion people that are undernourished chronically. There are, and I'll get the figures for you, I should have put this in, there are roughly, 2 billion people are iron deficient. Golden rice will improve the iron in the seed. Almost 1 billion are chronically undernourished. 140 million are suffering from vitamin A deficiency. The Helen Keller Foundation, which people feel has no axe to grind, is a partner with Erie to develop golden rice because they can see 140 million people don't have to be vulnerable to blindness. And so who's going to go up and say, we want more blindness? Now, you could do it by giving people vitamin supplements, but this is a much simpler way of doing it. But I don't think it's an agricultural problem. I think it's a science education problem. And you just look across the board how we have a smorgasbord of what science we believe, what science we don't care to believe, and there's no rhyme nor reason in any country that I've been in. It's a tough one.
Yep, Roger.
Roger, where am I? I know you're optimistic, and we probably are. Can we do it?
Okay, the question is I'm optimistic, and Roger's optimistic, and the short answer is, can we do it? Okay? The short answer is, you're asking agriculture to perform at a level that we have never seen before. When I look at the productivity gains that we need to have, the rate of increase, at the best of the Green Revolution, we were looking at 1%, 2%, maybe 3% per annum, probably more like 2%. And we were using a much lower base. 2% on 10 bushels per acre is 0.2 of a bushel. 2% on 30 bushels per acre is 0.6 of a bushel. It's a lot easier to get 0.2 of a bushel. Percents deceive you. Am I optimistic? I am, because I can't accept the pessimism. I'm not willing to ever accept the lifeboat. I didn't accept it at 18. I'm not going to accept it now. But I don't want anyone to underestimate what they're asking. And that is, we are facing an unprecedented asking to have rates of gain that only at the height of the Green Revolution, with a much lower base, were the percentages as good as what we're going to have to get in a rate of gain. It's going to be a challenge. And that's why, despite the end going off to the far lane, if you're going to go far, you better go together. And I think what we really should be seeing is the opportunity for agriculture as a whole, working in partnership with everyone. That's the best shot we've got. Are we going to make it? Boy, I don't know. But I guarantee you we'll be better off trying than if we didn't. And if we get closer, that's better than if we didn't get there at all. But I can't promise that, Roger.
Right over here in the back.
You had spoken or said something about, going back to Turkey. I wondered what you're looking for in going back to the older wheats.
Okay. The question was, in case you didn't hear it, when we went back to Turkey, what we were going back to look for. And the idea actually was this. It's kind of like the old Star Trek idea of parallel universes. So that you have wheats that are grown in Nebraska, heat, drought tolerance, winter hardy. And they've been developed in an isolated population. We do a lot of germplasm exchange, but we haven't done a lot with Turkey. In Turkey, you have similar climates. Heat, drought, severe winter hardiness on the Anatolian Plateau. And so they're breeding. And the question became, do they have some genes that by chance we don't have over here? And maybe we have some genes that they don't have by chance over there. Which, by the way, my predecessor actually released a line in Turkey. It so happened we were doing germplasm exchange. It was so good that they liked it and they released it. The line was Bolal. So we know that the climates are the same. But the question is are the gene pools the same? And if they're different, can I go to Turkey and get new genes in a background that already is winter hardy, heat stress, drought stress, all of those types of things? And it was looking for the new genes. And I apologize I didn't make that clear, but that's really what it was. It's to have two different gene pools that started together, potentially diverged. Can I go back, grab some new stuff, and bring it back? Thank you.
Please join me in thanking Steve for a wonderful lecture. Just a couple quick acknowledgments. I'd like to give a special thanks to our folks at Ed Media here who have provided the technical support for the lecture and getting it broadcast out. Thanks for that work. And to Judy Nelson, who is in the back, who has coordinated the lecture series so far, and we thank you, Judy, for your great work in getting us off to such a great start. We're in the mode of announcing our lectures 2 months in advance. So I'd like to draw your attention to the next upcoming lecture that will be December the 12th at 4 p.m. in Hardin Hall over on 33rd and Holdridge. Our guest speaker will be former Governor and U.S. Senator Bob Kerrey, who will be coming to give a lecture on conflict around the Missouri River, focusing on water. So an interesting year to give that lecture, as we know, with the year that we've had. And I'm pleased to announce that our January lecture, on January the 17th, it will be an evening lecture at 7 in the evening. It will be in Kimball Recital Hall on the city campus downtown. Our lecturer will be Mr. Steward Brand. Stewart is widely known as the father of Earth Day, that you'll recognize from the 1960s. He's the author. His most recent book is Whole Earth Discipline, and he'll be focusing on how agriculture is at the epicenter of sustainability. So we're very pleased to have Mr. Brand coming to our campus, and put that date on your calendar. Thank you for being here, for supporting the lecture series. Please come in December and January and encourage our students to be here as well. Good evening.